Genetics and Genomics in Medicine, Chapter 5, Single Gene Disorders, Inheritance Patterns, Phenotype Variability, and Allele Frequencies. 4. Allele Frequencies in Populations. Genetic disorders that are comparatively common and serious have somehow avoided being eliminated from natural selection. Why is this? Why should single gene disorders be comparatively common, but others very rare? In this module, we will be concerned primarily with allele frequencies and the factors that can influence them. Genes that are very large, such as the dystrophin gene, or that contain many repetitive sequences that confer structural instability, might be especially prone to mutation. Some of the most common single gene disorders, such as sickle cell anemia and the thalassemias, result from mutations in tiny genes. Even in some autosomal dominant disorders, a high incidence of the disorder may not necessarily mean that the underlying gene loci have high mutation rates, as we will learn later in this module. Some disorders may be caused by what is referred to as a selfish mutation. Achondroplasia is a common single gene disorder, but is caused exclusively by a mutation at just a single nucleotide, producing a highly specific change, glycine to arginine substitution at residue 380 in the FGFR3 fibroblast growth factor receptor type 3 protein. The nucleotide that is altered is not thought to be highly mutable. Instead, the mutation may promote its own transmission. Male germline cells that contain it may have a proliferative advantage and make a disproportionate contribution of sperm, so that there's actually a high allelic frequency, even though the mutation rate is not so exceptional. Interest is why certain disorders are common in some populations but very rare in others. Cystic fibrosis is particularly common in northern European populations, for example, and sickle cell anemia is especially frequent in tropical Africa, but virtually absent from many other populations. All of these considerations, what we mean by human population, is important. We could mean anything from a small tribe to the whole of humanity. An idealized population would be one with no barriers to random mating, but as we all know, mating is in practice far from random because of different barriers. They can be geographic barriers, cultural barriers, but mating is certainly non-random. This slide exemplifies how genotype frequencies are related to allele frequencies. In this example, we consider a locus A that has two alleles, A star 1 and A star 2, with respective frequencies P and Q. Genotypes are unique combinations of two alleles, one that arises from the father and one from the mother. In A, we can first construct a matrix of all possible pairwise allele combinations, whose frequencies are simply the products of the frequencies of the two alleles. In B, we then integrate any pairwise contributions that have the same two alleles, A star A2 and A star A1 is effectively the same as A star 2 A star 1 to get the frequencies of these three unique genotypes. So in that meaning that if you have a mutation on one particular allele and another mutation on another allele, it doesn't matter which one you consider first, but they're essentially the same thing. We'll talk a little bit later about something called the Hardy-Weinberg equilibrium law, and this will actually relate the genotype frequencies to the allele frequencies by binomial expansion equation mathematically accounting for these phenomenon. This slide shows a pedigree that exemplifies the use of Hardy-Weinberg equilibrium law to estimate disease risk. The Hardy-Weinberg law, or equilibrium, is a probability relationship between allele frequencies and genotype frequencies found in population under ideal conditions. So it is a mathematical relationship that assumes ideal conditions. And we just got done saying that there are not ideal conditions, but it is a mathematical way to predict what things would be under ideal conditions, and then one can go from there. If we take the example in figure 1, the healthy proband, which is arrowed, has a sister with cystic fibrosis and is worried about the prospect that he and his wife might have a child with cystic fibrosis. His wife is Irish, and the Irish population has the highest incidence of cystic fibrosis in the world and affects one birth in every 1,350 individuals. The proband's parents can be assumed to be carriers, each with one normal allele and one mutant allele. Because the proband is healthy, he must have inherited one of 3 possible combinations of parental alleles. N from both parents, homozygous normal, N from father and M from mother, and M from father and N from mother. So from Mendelian principles, he has a risk of 2/3 of being a carrier. The risk of his wife being a carrier is the same as the probability of that a person picked at random from the Irish population is a carrier. If we assign a frequency of P for the normal allele and Q for the cystic fibrosis allele, the Hardy-Weinberg law states that the frequency of affected individuals will be Q squared and the frequency of carriers will be 2PQ. Because population surveys show that cystic fibrosis affects 1 in 1,350 births in the Irish population, one can mathematically calculate what the probability. So the combined risk, if one calculates this out using the formula described in your textbook, is that both the proband and his wife are carriers, and that there is an overall risk of 3.5%. Remember, we said that the Hardy-Weinberg law applies to an ideal population. And of course, we don't exactly have ideal populations. One instance is that we really do not have non-random mating. And then there are other instances that we can use to explain. And this slide actually shows random sampling of gametes. And that when one randomly samples gametes, you can see that clearly Hardy-Weinberg equilibrium is not in play. So what can account for that? Well, there are a variety of factors, and one is purifying negative selection. And we talked about this earlier in the course. Purifying negative selection refers to the fact that if a person is affected by the genetic disease, it is most likely going to affect his ability to reproduce. And so one can start to move out that gene in the population. In addition, we have the notion of new mutations. So new alleles are constantly being created by factors within cells that cause a particular gene to lose its function. And so there are new mutations that skew the Hardy-Weinberg equilibrium in a different direction. Another factor can be influx of migrants. So if a population absorbs a large influx of migrants with different allele frequencies, this is then going to affect the gene pool or the pool of alleles in the population, again skewing the Hardy-Weinberg equilibrium. And so as this slide exemplifies, if we do a random sampling of gametes, we can tell whether these factors are at play and whether or not there's a genetic, if we sample over time, we can detect the phenomenon of genetic drift. And genetic drift is ways in which we can observe in particular smaller populations, the allele frequency changes in a given smaller population. This slide exemplifies population bottlenecks and founder effects. So this is another way that we deviate from the Harvey-Weinberg equilibrium, and what are things that are driving genetic pool change. So genetic drift is the most significant when population sizes are small. There have been several occasions during our evolution when the human population a population bottleneck. As a result, genetic variation in humans is much less than in our nearest relative, the chimpanzee. Another type of population reduction has periodically happened during human migrations. Again, in a small population would represent a subset of genetic variation in the original population and have different allele frequencies. When we have a small group of individuals that immigrate to form a separate colony, this would lead to a new genetic population, and they would have limited genetic variation in allele frequencies that reflect those of the original settlers. And this is referred to as the founder effect is a very real effect. And this table shows examples of single gene disorders that are common in certain populations because of a founder effect. So, for example, we have aspartyl glucosinurinemia, which is common in the Finnish population, and has a carrier frequency of 1 in 30, which is quite significant. If we look at Ellis-Van Creveld syndrome, that is very prevalent in the Amish population in Pennsylvania and has a carrier frequency of 1 in 8 individuals, and it's traced to a single couple who immigrated to Pennsylvania in 1774. And it goes on. We see this happen in the Ashkenazi Jewish population, in Puerto Rican populations. Alzheimer's disease type 3, early onset, occurs in remote villages in the Andes, and all descendants from a couple of Basque origin who settled in Colombia in the early 1700s. And so it is quite interesting about the founder effect. Finally, this slide shows the concept of relative fitness, or F, which is a measure of the reproductive success of a particular allele to persist in the population. And it uses a scale from 0 to 1 to rank the capacity of individuals to reproduce and have children who survive to reproductive age. So thus a fitness of 0, which is genetic lethal, means a consistent failure to reproduce, and some mutant alleles are not transmitted vertically to their descendants. Loss of mutant alleles from the population by purifying selection is balanced by the creation of new mutant alleles by fresh mutations, keeping constant the disease allele frequency in the population. And this particular slide shows for different situations autosomal dominant, autosomal recessive, and X-linked recessive disorders. Sickle cell anemia is a classic example of heterozygote advantage. And so sometimes a heterozygote advantage refers to when natural selection favors carriers of recessive diseases. So sickle cell anemia provides this example because it's very common in populations where malaria caused by this Plasmodium falciparum parasite is endemic. And so you run a high risk of being infected by that parasite. And in some malaria-infested areas of West Africa, the sickle cell anemia allele has reached a frequency of 0.15, far too high to be explained by recurrent mutation. Now, sickle cell heterozygotes have red blood cells that are inhospitable to the malarial parasite because of their abnormal shape. However, the heterozygotes have a higher fitness than than both the normal homozygotes and the disease homozygotes. The normal homozygotes often die from malaria, so they don't reproduce. The disease homozygotes also may die because of the sickle cell disease, but the carriers themselves, which has allowed this, which otherwise, when present in a homozygous state, would be lethal, has allowed this allele to persist. So it is very interesting and important to consider the heterozygote advantage and its effects on natural selection.